it's a great pleasure for me to be here in San Diego. First of all, this is an amazing place for stem cell research. All of California is amazing. This is a mecca. And San Diego in particular, this is an outstanding institution that you all have built. And it's the first time for me to be here. So thank you for showing it to me. And it's very inspiring. I'm staying actually in La Jolla Coves, and that's the first time I've ever been there. And I was fortunate yesterday to be able to go out, and it was a sunny day, and I could look out, and the wildlife is unbelievable. To see the sea lions diving and the birds, by the hundreds lined up on the rocks and flying around. And it's just so serene and so magical. And, for me as a biologist, when I see nature like that, it's very inspiring as well. And I think it's just hard not to believe that anything is possible when you see that. So the title of my talk today is Modeling Kidney Disease and Regeneration with Organoids Derived from Human Pluripotent Stem Cells. And these are just a few disclosures. And I wanted to start with the rationale for why kidney disease can benefit from stem cell research, why we need stem cell research. And there's really two major arms here. The first is that the kidneys have no clinically significant ability to regenerate lost tissue mass. So as pieces of tissue are lost, there's no way for the body to grow them back. And although we have modalities to partially replace kidney function, these modalities are limited and they have severe side effects. The second rationale is that we don't understand well how the different disorders that cause chronic kidney disease actually work at a root and molecular level. And as a result, we don't have medications that prevent the disease early in its progression. And one of the big challenges is that kidneys are so complex. Each of the kidneys is made up of about 1 million of these filtering units called nephrons. And these are not just passive filters. They exquisitely balance and take back into the body all the nutrients that we need at just the right concentration so that the blood stays constant. And this is it has many different cell types and there's many different functions that are involved. And trying to recreate that biologically is a major challenge that has stymied the field for many years. But we need to do that because this disease is a major health problem. And we actually spend more on kidney disease than on our entire NIH research budget every year in the US. And 1 out of every 10 people will have kidney disease. So we do need to make innovations here and better understand and utilize stem cells. And as this audience can appreciate, human iPS cells present a paradigm for doing this. It raises new possibilities that never existed before, so that we can take cells from our own bodies and reprogram them back to an early embryonic stage before any of the organs have formed, including the kidneys. And we can now give ourselves back that ability to grow kidney tissue and use those structures that form, which we call organoids, for studying different disorders in a petri dish, but also for potentially transplanting back into our own bodies where they would be autologous and would not require immunosuppressive therapy and could be generated on demand. So for today's talk, I'm going to hit on 3 major topics. First, I'll briefly introduce the kidney organoid and its origins. Then I will talk about its applications for studying disease in a dish, using polycystic kidney disease as an example. And finally, discuss the longer-term prospects for using these types of stem cells for regenerative medicine. And in terms of the biogenesis, we appreciate that the nephron structures that I showed you before differentiate from specialized progenitor cells, which we call nephron progenitor cells, that arise during embryogenesis. And these then differentiate through a mesenchymal to epithelial transition into the different types of epithelial cells that are present along the nephron unit in a proximal to distal axis. And this occurs shortly after the germ layers are actually established. Now, when I started in this field, we did not have a way to turn iPS cells into kidney, and we had no idea what it would look like. And as a postdoc, I studied this challenge, and what we discovered is that if you just take pluripotent stem cells and culture them in a thin layer of matrigel in 3 dimensions, they round up into spheroids. And then if you treat them with a high dose of a GSK3 beta inhibitor called Kyre, they would spontaneously differentiate into the renal lineage. And the way we knew that this was happening is because you could actually see these tubules form. If you look here on the right, you can appreciate the formation of these mounds of tubular structures that were not present normally in any other type of differentiation. And this occurred over about 3 weeks, and when we then proceeded to examine these for different markers of the types of cells present in the kidneys, including the podocytes, which are the filtering cells of the kidney that interact with the blood, as well as proximal tubular cells, which are just downstream of the podocytes and take nutrients back into the body, we found that these cell types were expressed in very well-differentiated forms, and they were very distinct in a way that we were unable to achieve using other attempts to differentiate. So they compared very favorably to the orientation and elongation of these structures in actual kidney tissue. Not a perfect replica by any means, but it's still rather mind-blowing to consider that this organized structure that looks almost like a kidney was once a skin cell in its past life and has now been completely reprogrammed. And we now appreciate that these organoids contain many different types of cells that are present in the nephron, including epithelial cells, in addition to the proximal tubules and podocytes. There are distal tubules and parietal epithelial cells, which line the podocytes and form these capsule-like structures, similar to what we see in actual kidneys. And altogether, there's about 11 different cell types. And beyond the epithelial cells, there are stromal cells that arise. And these, we believe, have an identity that is related to the actual organ. So there are endothelial cells, blood vessel cells that form and begin to interact with the epithelial cells in a manner that is reminiscent of what happens in the actual tissue. So they make pretty pictures, but can they do anything? This is always an ongoing question for us in this field. What we have observed is that the tubular structures in the kidney organoids have an ability to perform solute transport. And I'm showing you a couple of examples of this. The first is in a microfluidic device where we're passing glucose and dextran, which are labeled in green and red, over the organoids. And as these solutes pass by, the organoids transport them into their cells and into the lumens of those tubules. And that's the type of thing we might expect a kidney to do. The second example is shown below. This is a forskolin-induced swelling assay. So each of these little blobs here is a kidney organoid, which is adherent in our case to the Petri dish. And when we add in cyclic AMP or forskolin, which stimulates its production, we see those tubules swell up and they form these little water balloon-like structures, which we call cysts. And that's indicative of a solute transport event, which is followed by water flowing into those tubules and expanding them. So they do have rudimentary kidney functions. We would certainly like to see additional functions, and we expect that as the field progresses, there will be increased functionality of these structures. So that's the short primer. And now I'd like to discuss the applications of these organoids. And I'd like to start with polycystic kidney disease. This is a fascinating disorder in which the tiny tubes within the kidneys and also other organs swell up, similar to that last slide that I just showed. And they form these expansive sacs of fluid, which are called cysts. And there are many cysts within a polycystic kidney. That's really diagnostic of the disorder, as you can see here. And as this occurs, it crowds out the healthy tissue such that by middle age, these patients need a kidney transplant. Now, we've known for 30 years which genes cause this disease, and these are two genes. One is PKD1 encoding PC1 or polycystin-1, and one is PKD2 encoding polycystin-2 or PC2. And we know that these encode a channel receptor complex that localizes to the primary cilium, which is a hair-like appendage that sticks out of cells. But we don't understand actually what this channel receptor complex does and why, when it's dysfunctional, you have this cyst phenotype. And one of the real challenges is that if you try to take cells out of a patient with PKD, they don't form cysts in a PKD-specific way. So there was no in vitro model of this disease before the kidney organoids. So we wondered whether the organoids might actually unlock the phenotype of PKD because they're three-dimensional. And indeed, that is the case. If we generate cell lines, iPS cell lines, in which PKD1 or PKD2 have been removed, have been knocked out, we observe that the organoids form initially normally, but then go on to give rise to these large, expansive, fluid-filled cysts that you can see here by the naked eye. And this is a movie of what this looks like over here on the side. We'll see if we can get that to play. So you can see here the size that these cysts arise. They start off as tiny organoids, 150 microns in diameter. And over a period of weeks and then months, they take up fluid and continue to grow and reach centimeter diameters. And another thing that I think is really fascinating is that if you look at the media in which they're growing, it's a different color from the fluid inside the cysts. And that's because the phenol red, which is in that media, is getting transported out of these cysts. So they're still continuing to perform a transporter function. Now, what's really remarkable is that this phenotype arises specifically in the knockout lines, both for PKD1 and for PKD2, but not in isogenic controls. So we know that this is really PKD in a dish. And this teaches us fundamentally that cyst formation can be recreated just with cells in a petri dish. We don't need the rest of the body. There's something really basic about this disease. So we've studied this over the years in many forms, and I won't go into all of the studies, but I wanted to highlight work by an MD Ph.D. student in our lab, Courtney Vishy, who recently graduated. And Courtney took on this massive cloning project to generate organoids that have clinically relevant nonsense mutations. And there's no animal out there with this type of nonsense mutation, but it's extremely common in patients. And she generated, using CRISPR base editing, 4 different types of mutants, each of which has a truncating mutation in either PKD1 or PKD2. And this is just over here a schematic of all the different clones that she generated. Interestingly, in addition to the homozygous clones, which were knockouts, we also got a very nice set of heterozygous clones, which we were able to study as well. And just one of the experiments, and this is a recently published paper, that was quite interesting is we were able to use the opposite type of base editing on these mutants to rescue PKD cyst formation. So in this case, we've started with a mutant that generates cysts, as you can see, over a period of a couple of weeks. If we then use a base editor in those original stem cells, we can create a heterozygous mutant, and that mutant has no cyst phenotype. So this teaches us that even just restoring part of the protein, like half of the genes that are dysfunctional, is sufficient to completely rescue the cyst phenotype, which you can see here on the right, quantified by cyst area at 0, 7, and 14 days, as well as the percentage of organoids that form cysts, which is about 75% in the mutants and below 20% in the ones that have been gene corrected. So this was the first time we've ever rescued the PKD phenotype using a genetic approach. But to do gene therapy would be very challenging and is going to take many years to develop. So we wondered whether there might be a chemical approach, a pharmaceutical approach, that is more ready for the clinic. And we reached out to a company called Elox, which was generating these very interesting class of molecules called eukaryotic ribosomal selective glycosides. There's two things that are important about these. The first, they allow read-through, a degree of read-through of premature nonsense mutations, such as the ones that Courtney had generated in these cell lines. So whereas the ribosome would normally see a stop codon and disengage, in the presence of these glycosides, the ribosome is stabilized and in a small percentage of the reads capable of generating a full-length protein. And this is specific to the premature nonsense mutations. It doesn't affect normal nonsense mutations. The other interesting thing about these drugs is that kidneys naturally clear glycosides from the body, which means that these drugs get concentrated in kidneys, and that makes the kidneys a nice potential site of action to target. And there are no animal models that have these types of mutations, so the PKD organoids that Courtney had generated were the only option we had to see whether they might be useful for PKD. And to make a long story short, these read-through therapeutics do a very nice job of blocking cystogenesis, both in our PKD1 and PKD2 mutants with nonsense mutations, as shown here. So as we increase the dose of these compounds from 10 up to 90 micromolar, we see a dose-dependent decrease in the cyst area in all of our mutants, which was statistically significant. And we also observed this not only with the flagship drug that the company is working on called ELX-02, but also with a newer compound called ELX-10. And this was very consistent among our mutants. So we believe that this is potentially a therapeutic approach for PKD that could be useful for a large class of patients with these types of mutations. Because these are drugs, they could be fast-tracked to the clinic, and we know that they would be safe at particular doses in patients. So we think this is a very promising approach. So my conclusion from this first part of the talk is that although the model might not be perfect, it is still could be useful. And if we think creatively, we can find ways to move therapeutic applications faster into a clinical setting using organoids and using the information they provide. And we've recently begun a 5-year project to develop mini kidneys as DDTs, or drug development tools, which would be qualified by the FDA for this type of drug development. And there's a recent passage through Congress of the FDA Modernization Act 2.0, which allows for drug developers to gain market approval for a drug using methods other than animal testing. And this is a nice example of how that process might be able to work. But in order to really get there, we will need to work with the FDA and with regulators to make sure that the systems are robust enough and reliable enough to give us the types of readouts that would advance molecules into clinical trials. So I think that overall organoids fit in at many different stages of drug development, from the discovery stage, where you could be testing out many different molecules, just trying to hit a needle in a haystack, all the way to the stages of repurposing molecules that are already known drugs for particular conditions, such as polycystic kidney disease. And hopefully this will lead to drugs for this disorder, which has currently only one approved drug, and that drug is available only to a minority of patients due to its side effects. And one of the other lessons I've learned going through this process is that commercialization can help us create these types of therapies. We're seeing increased interaction between academia and industry. I've been part of 2 such efforts. One is the development of a stem diff differentiation kit with stem cell technologies. So they've taken the protocol that I developed as a postdoc and commercialized it so that anyone can differentiate organoids in their own lab. Another is a recent spin-out company that I've helped found together with the University of Washington Co-Motion office, which is named Pluripotent. And Plurexa seeks to take pluripotent stem cell technologies and the ability to multiplex and make these technologies available, particularly for kidney disease, to a wide range of investigators. And I think that by having these types of commercialization efforts, we can really take the basic research discoveries and move them more quickly into the wider marketplace. So now for the rest of the talk, I'd like to discuss the potential of these organoids for regenerative medicine in the kidney, which is kind of the holy grail of the field. And here we recognize that there are certain patient populations for whom a drug will not be adequate. And an example of this are patients with cystinosis. Cystinosis is a disorder of cysteine accumulation. So it has nothing to do with cysts. It has to do with the amino acid cysteine. And this amino acid normally accumulates in our lysosomes and is exported rapidly into the cytosol by cystinosin, which is a transmembrane transporter. And when this transporter is broken, as it is in this rare disorder, the patients have a constellation of medical challenges. And the kidneys in particular are very sensitive to cystinosis, which results in a need for kidney replacement therapy early in life, typically in their second decade or third decade. So for patients with cystinosis, there already is a drug, and it's called cysteamine, and it can help ameliorate some of the symptoms of cystinosis, but the patients still need to have kidney transplants. It's not adequate, and they're going to need multiple kidney transplants in the course of their lives. And as I mentioned, fundamentally, what appears to be happening is that the kidney tubules and proximal tubular cells are atrophying due to an accumulation of cysteine, and this actually crystallizes inside these tissues. So you can see these cysteine crystals by histological examination. And as a result, the nephrons deteriorate and there's declining renal function. And these patients would benefit greatly if we had a way of giving them back additional kidney function. There's hope. One of the really inspiring stories that I saw this year is that of Rick Slayman, who was a patient with kidney disease and very bravely opted to have the first pig kidney transplant in a living patient. And this is an amazing story, really, where you have a gene-edited pig kidney that has gone into a person, and the gene edits were made to make this kidney more tolerable to that individual so that there wouldn't be an immediate rejection. And so far, so good, as far as I understand. So this has really gotten a lot of attention and could be a Nobel Prize-winning experiment, I think, in the end of the day, if it's successful. So I think this is an inspiring example of what we can hope to see in the future for kidney medicine. But those types of pig kidney transplants probably won't be the silver bullet for folks with cystinosis. We understand that these xenografts will get rejected by the body eventually. And these are some data from experiments that were performed in monkeys with these types of xenografts, where although the monkeys were able to survive post-transplant for much longer, thanks to the gene editing, the extensive gene editing in these kidneys, they were eventually rejected because they were not monkey kidneys. So these types of xenografts will require strong immunosuppression and monitoring, and they will eventually be rejected. There's also questions regarding how well they're going to function, given the differences between mini pigs and humans. And there's some potential for zoonotic infection. So I think in the near term, at least, that technology will be limited to humane use. But it really can make us think about the potential advantages as well as the limitations of iPS cells. I think iPS cells are kind of the yang to the yin of the pig kidneys. iPS cells are our own cells. So in terms of tolerance, they're going to be the opposite of xenografts. They're derivative, they can grow forever, so we can really scale these up. They're a potential resource. They would not need any immunosuppression. But Unlike the xenografts, we can't grow whole organs from iPS cells yet. And we haven't even tested them in animals for transplantation for a wide variety of organs and where we would need replacements. In fact, it's amazing when you look at the number of actual clinical trials that have been done in the world from iPS cells, the number is something like 30. It's not very high. So there's still a long way to go. We're really at the tip of the iceberg for this technology. So we went about with support from a foundation called the Cystinosis Research Foundation, going about creating iPS cells from people with cystinosis. And I wanted to show how this is done. We provide the individuals with a collection kit, and they provide a urine sample. And from that urine sample, which is then returned to the lab, we can generate urinary cells. Now, these are not stem cells, but we can reprogram them into iPS cells using the Yamanaka factors. And now we have the ability to make new tissues for these patients. And obviously we're interested in the kidneys, but these could be used for any organ of the body. So we've done this now for 19 patients, and we have hundreds of vials in the liquid nitrogen generated from these individuals. And because this is a long-term study, we can keep in contact with those patients in the future. So if the cells ever become useful, we can then recontact the donors. Using these, we've found that the iPS cells from patients with cystinosis can survive, they can proliferate. They're really very similar to iPS cells that don't have cystinosis. And they can turn into kidney organoids in a plate, which don't have any obvious defects. So just like the patients, cystinosis is not a barrier to the development of the kidneys. And they even express the types of transporters that we would normally associate with mature kidney organoids. Now, for years, we've been interested in knowing what happens when we put organoids back into an animal. And this is some early experiments that we've published where we've taken the organoids after we've grown them and implanted them beneath the capsule of the kidney. So just on the periphery of the kidney, there's this nice little capsule, and you can slip cells between the cortex of the kidney and the capsule and let them grow for several weeks or even months. And these are some of the early data that we observed. We saw that the organoids would grow inside this kidney capsule. This is human nuclear antigen, so these are all human cells here. But there's a small proportion of mouse cells that are arising within the graft as well. And these are the vascular cells, and these, interestingly, are capable of growing into the podocytes, or the filtering cells of the kidney. So the human podocytes are surrounding a small proportion of mouse vascular cells in this study. And that's the beginnings of what could be a filtration barrier forming. But it wasn't very robust in this situation. So as I've mentioned, the nephrons normally differentiate from nephron progenitor cells, and we suspected that this process is also happening in our Petri dish. And this then gave rise to the question of if we could then implant the progenitor cells instead of the mature organoids, would it lead to a more robust graft? And this was a project that was taken on by Thomas Vincent, who's a bioengineering Ph.D. candidate in our laboratory. And one of the first observations that we made is that there is a layer of progenitor cells present in the organoids and around the 9th day of differentiation. And it forms a capsule just above the differentiating wave of cells as they turn into tubules. So this is a staining of NCAM, which is a progenitor cell marker, and lotus lectin, which is the tubular cell marker. You can see this lower wave of differentiation that's occurring beneath these green cells at the top. And this is very reminiscent to what the kidney looks like, where you see this, in this case, a yellow layer of cells just on the edge of the kidney and this descending wave of differentiation below it. So Thomas went on to analyze different markers that we associate with nephron progenitor cells, and in doing so, he found a window of time in which these markers were highly expressed. And this indicated the presence of these nephron progenitor cells in the organoid cultures. And when Thomas implanted these nephron progenitor cells into the mice and waited 3 weeks, we observed something remarkable, which is that the mouse kidney sent in blood vessels that amazingly vascularized these organoids and began to form structures that really look like filter units. And you can appreciate this here. The green is the mouse vasculature. It's invading these red human podocytes and forming what we would call a capillary tuft inside it. That's a tortuous structure in which there's an inlet and an outlet, and blood can pass through and potentially even be filtered through these structures. And we observe in this image the surrounding of this tuft by human parietal epithelial cells and an expansion of what we would call a Bowman's space, where the blood is passing through these capillaries. It's being filtered by these podocytes in purple and then entering a space in which the larger molecular weight proteins and blood cells have been excluded so that we can create a very primitive form of urine. This is quite remarkable that it occurs as well as it does, considering that there's a huge species barrier between human and mouse, and that this is an adult mouse that's sending in blood vessels. Yet there's something about this developmental program of creating a glomerulus that dominates and enables this to occur. And what it means for us is that we can envision putting in stem cells and having them become vascularized by the body to form new nephron subunits. Another remarkable aspect of these grafts is that they contain mesangial cells, and these are supportive stromal cells that are normally in the glomerulus, but which we don't see in our organoids in the dish. And these mesangial cells are essential for supporting the endothelial cells, and they actually allow that curvature and looping of the capillaries that permits the glomerulus to form normally. So the presence of these cells is a very good sign for the potential of the kidneys to the grafts to potentially function. So just a couple more details about this experiment. Getting back to cystinosis, we've been doing these experiments in immunodeficient mice that have no disease, but recently we've generated a mouse that is both immunodeficient and has cystinosis. And these are some of the cysteine levels taken from the organs of these animals, the kidneys, the livers, and the hearts, compared to the wild type animals, which are isogenic. And you can appreciate there's a dramatic accumulation of cysteine within the kidneys of these animals. So we're breeding these now as host mice, and we hope that they will be a model for the cystinosis patients that we eventually hope to treat and would need kidney transplants. The other aspect of it is that we have an opportunity with the iPS cells to correct the genetic mutations that would cause the disease before we would do the cell transplantation. And to model this, we've used CRISPR-Cas9 and developed a quote-unquote off-the-shelf method to knock in the wild type CTNS gene into chromosome 19's safe harbor locus. And we do this in the iPS cells and screen them, and we can pull out clones. And these clones, when we add doxycycline to them, will induce expression of the cystinosin transgene. And then we can really robustly detect it at levels which are clearly at or well above even the wild type levels of cystinosin in a patient. And this also has a significant decrease in the accumulation of cysteine in these cells. So the path that we see going forward is one could derive iPS cells from a patient, edit the genes, and then use that as a source of transplant, which would be an autograft, instead of the current paradigm of an allograft with immunosuppression. So a humble urine sample can be the start of something big, we hope. Nephron progenitor cells, we now know, can differentiate inside a living host, and it really helps to be doing this with early progenitor cells as opposed to the mature organoids, which are probably already too far gone in terms of their developmental process to form those types of glomeruli that I was showing. And the grafts can recruit endothelial cells from the host to form those glomeruli-like structures, which is, I think, a mechanism that we can further explore as we develop kidney transplantation therapeutics. Still a very long way away. I would not encourage anyone to try this at home or even for patients to try this. We need to test out these therapeutics in mouse models and develop them properly to make sure that they work and that they're going to be safe. So this is going to take years, but I'm hopeful for the future of iPS cells and nephron progenitor cells as a potential therapy. So that's all I wanted to present. I would like to thank the people in my laboratory who have contributed to this work. Here's a relatively recent picture. It's a really great group of people. We have our own little niche. I think that research requires a niche, just like organs require a niche, and we nurture each other, and then occasionally we develop something interesting. And similarly, part of our niche is really a very helpful and supportive network of organizations that contribute to our research and make it possible. So thank you for your attention, and I'll take any questions.
Thank you so much for your amazing work. I was just wondering, in the part 1 of your presentation, when you use a glycoside, does it just skip the nonsense base pair and the rest of the protein is perfectly normal, or is there like a frame shift? What's
yeah, so it brings in a cognate tRNA, essentially. So instead of having a stop codon, there's another amino acid that comes in,
so you get
in a small proportion of the cases. And so the ribosome makes a mistake. Yeah.
Okay, awesome, thank you. And that doesn't necessarily cause problems with the function of the...
Well, you know, I think it's better than losing the whole protein.
Yeah.
It's not as maybe ideal as having, you know, the original sequence, but it's sufficient. You know, in many cases, these mutations are just a single base pair anyways. So tRNA might actually give you back the original protein.
Great talk. I just had a quick question about the differentiation aspect of it. Obviously, the protocol you've developed pretty much from scratch, which is very impressive. Have you seen any off targets, or like the levels of which you get off targets in vitro, and also when you put the progenitors in vivo? Obviously, you mentioned that there is still continued differentiation. Do you see anything that isn't non-kidney related?
Yeah, great question. You know, you've really hit on an important point here, which is that we do have difficulty in controlling the differentiation of iPS cells into a specific lineage. And in the case of the kidney, I think the major off-target that we see is neurons. There seems to be a neuronal component that's differentiating in the dish side by side with the organoids, the kidney organoids. It's almost like a little brain organoid that forms next to it. So we haven't been able to get rid of it. There is another laboratory that of Ben Humphreys, which has done an interesting experiment where they were able to reduce that off-target population by treating with certain chemical compounds during the differentiation process. So yes, I think that's important. The other aspect of it is in vivo, we don't know yet all the different types of off-target populations that form. But we do observe, if you let the grafts go for long periods of time, we do observe cartilage appearing in the grafts. So the off-target in vivo may actually be a little bit different from the off-target in vitro, but there is also an off-target. And we need to control that because those cells continue to grow beyond the point where the kidney cells grow. So we have to find ways of controlling the differentiation better. And I think that's been just a major barrier in the field in general.
Thank you.
Hi, thank you for this insightful talk. On a similar note and kind of a similar difficulties for progenitor and stem cells, have you monitored these grafts long enough to see if the progenitor cells would form some sort of tumors or uncontrolled proliferation?
Yeah, we haven't observed it in the grafts. There are forms of tumors that can occur when kidney progenitor cells have mutations in them, such as the Wilms tumor, and that's essentially uncontrolled growth of the nephron progenitor cells. But our stem cells actually have the opposite problem. They don't seem to be able to grow even for multiple rounds the ways that we would hope them to grow. So I think for now, that piece of it is less of a concern. But you're right, we need to keep an eye on that, and we need to develop assays for a situation where we might get uncontrolled growth.
So I was wondering, I work with brain organoids, and I do see the same cysts, maybe not as extensive, but very similar cysts in wild-type lines of brain organoids. And they've been suggested that they're choroid plexus and that they're functional. I'm wondering if you separate the diseased organoids from each other instead of keeping them in the same well, do the cysts grow as extensively as they would otherwise?
That's a good question. So they certainly grow. So we do this now more routinely. We grow them in 96 well plates and as individual organoids, and they still give rise to these cysts.
Okay.
So I don't know, though, if they would get quite as big because they don't have as much space in those tiny little wells. But I think if you were to grow one in a 6-well low attachment plate, like the one that I was showing, they would probably get very large. So, there's a lot of things that form cysts. There's many different ways to form a cyst in biology. It's sort of a default structure. So what's interesting in this case is that it's really the mutants that form the cysts and not the controls. And that's how we know this process must be the disease. But there's probably some similar mechanisms as general cyst formation, for example, what you're seeing.
Yeah, I think it's interesting to think about because I find that if I have one brain organoid with a cyst on it in a well, within a week or two, most of the organoids will then have a cyst. So I wondered if that might contribute at all to
I
the disease
see,
progression.
yeah. Well, that's an interesting question, and maybe we should talk. There might be a growth factor there.
Let me while we take the next in-house question, let me take one from online. Diabetes is a very common cause of kidney disease. Are any of the approaches that you've explored applicable to diabetic kidney disease?
Yeah, that's a great question. I've actually got a slide I'll put up here related to that. I didn't have time to go into this in the talk. DKD, diabetic kidney disease, is maybe the major cause of kidney disease, and we don't have good models for it in animals. So we've been working on developing a model of DKD in the organoids, which is shown here on the right. And we believe that the podocytes in the organoids actually show a dysfunction in the presence of high glucose and undergo a process of effacement and migration out of the tuft. So that's work that's still ongoing. It's not published, but we're very excited about it. And hopefully it will create a model for understanding that mechanism in our bodies and a place where we could screen for new therapeutics for diabetic kidney disease.
Thanks, that was very cool. So I wanted to ask a bit about the genetics background. So for PKD, so it sounded like this is all loss of function. There's no gain in function in both diseases that you described. Is that correct?
That's correct. Yeah, these are both both PKD and cystinosis are loss of function disorders. PKD, there's a wide spectrum of mutations, but they seem to be loss of function. Again, we don't know exactly what the function is of the proteins, but
Right.
because there's
so
so
there
many.
could be some gain of function, like toxic.
In theory, it's possible that there's a gain of function element to it, but the genetics data points strongly towards it just being a general loss of function in the pathway.
So if did anybody try to just crank up the gene and that would ameliorate this, or?
In the mice, you can crank up so there's actually an experiment in mice recently where they've re-expressed polycystin 2 in a polycystin 2 knockout. This was work done by Stefan Somlo's lab at Yale. And really cool results where they actually saw the kidneys get smaller and the cysts get reduced. They saw a therapeutic effect simply by overexpressing polycystin 2 at certain stages of the disease. However, if you overexpress PKD2 or PKD1 too much, you also get some cyst formation, which is a little confusing. And that's work that's come out of other labs over the years. But I think that overall, it would be very interesting to do those types of overexpression experiments in the organoid model as well, and see, what's the dose that's actually safe and helpful. Yeah.
And is there overlap in redundancy? So patients that lost PKD1 or PKD2, they have the same symptoms, same severity, or?
So the PKD2 mutants, the PKD2 patients have a less severe course of disease. Why that is not really clear yet mechanistically. It could be, though, that the patients accumulate secondary mutations at a lower rate. So it's not really clear that PKD2 is any less important than PKD1. Very interesting questions. In our organoids, we have not seen a difference in the rate of cystogenesis in the PKD2 knockouts versus the PKD1 knockouts. And we've also seen a lot of interactions between these proteins. So if we knock out PKD1, we actually see a degradation. If we knock out PKD2, we see a degradation of polycystin 1. And if we knock out polycystin 1, we see problems with polycystin 2 localization to the cilium. So I think they're very joined at the hip, and that complex, you need to have both components of that complex to do whatever it's doing to prevent the cyst from forming.
Great. From the Zoom, this questioner was interested in the model in which you implanted a kidney made from organoids. What kind of urine did the animal make, and did you characterize it?
Yeah, we have not yet characterized the urine. It's going to be down. The kidneys in those animals were mouse kidneys, and they were fine. So I'm sure most of the urine is just mouse urine. But it would be interesting to look and see if there's any human proteins, for example, in the urine. We don't know.
All right, one question coming on Zoom is, what are your thoughts about, after making the organoids, dissociating the organoids, purifying them into specific cell populations, and implanting those? Or do you think it's important to actually have the organoid structure for integration and function?
Yeah, it's a great question. I think it depends on the disorder. If it's a disorder where there's only one cell type that's really deficient, and you could repopulate it with a purified population, then that's a more logical way to go than to try to grow a whole new nephron. And it gets at that question about controlling the cells and the off-target populations, right? Because if you can purify the descendant cell type, then that's going to be less likely to give rise to an off-target population. So it's going to be a safer therapy. So I think it's a great idea. For other disorders, we may just want to grow new nephrons and take full advantage of the iPS cells' amazing abilities, right? So we'll have to see where the technology goes. If we can control that process well enough, then that will be more of a off-the-shelf remedy.
Another one that came online is actually an interesting question. Have you looked at hormone secretion from your kidney organoids to verify the synthetic function of kidneys?
Yeah, that's a good question. We haven't looked at the hormone and the synthetic function. We've been really sort of tunnel visioned on the filtration and more classic functions of the kidneys, but the kidneys do a lot of other things. They make EPO, and they make renin, and we've not really examined any of those functionalities. Some of those cell types we've tried to find, and they're more niche cell types, so they may only arise actually in vivo, not in vitro.
So the kind of experiments you haven't gotten a chance to do, for example, condition medium from the organoids and see whether they cause a blood vessel to constrict, or cause hematopoietic stem cells to erythroid progenitors to proliferate or anything.
Yeah, there's a bunch of labs though that are working on these questions. So we've got to sort of we've got to focus a little bit on what we think is most interesting to us, but thankfully the field has really taken off. So there are some interesting publications already about the synthetic activities. And doing this type of experiment that you're describing is great. I mean, this is what's so fun about the organoids. You can ask these questions that you just couldn't ask before. And we have no idea what the answer is going to be.
One more question coming on Zoom was, when you talked about pulling out the renal progenitor cells and putting them back in, did they need a scaffold support in order to engraft, or can you just send them in as a cell suspension and engraft?
I mean, I'm hoping that they can just go in as a cell suspension. So far we haven't used any scaffold. We've played around a little bit with implanting scaffolds, but that kidney capsule is pretty tight in the mouse for getting a large structure or a scaffold in there. But it is definitely worth thinking about whether a scaffold could enhance certain types of transplants. And the kidney capsule is probably not an ideal site for many of these transplants ultimately. So that's something we've got to figure out, is what is the best way to put the implant in.
Any other questions in the room? I'm not seeing any other questions on Zoom, but I'll maybe then end with take chairman's prerogative and ask just one last question. And this is more a philosophical question, so I know you won't have the answer. But this notion of taking human pluripotent stem cells, whether they're iPS or ES, and sending them down a particular lineage to make an organoid, whether it's gut, lung, brain, kidney. Very often we will see, for example, kidney is mesoderm, gut is endoderm, brain is ectoderm. We think we use a protocol that should give us pure endoderm or pure mesoderm. Nevertheless, we get what we want, which is a functional organoid, but then there are other germ layer derivatives there. And sometimes we use the pejorative term and call them off-target effects. The other way of looking at it is that we actually do need these other germ layer derivatives to get a functioning organ, because that's actually how the vertebrate body plan is made. It's not a pure mesoderm. So what are your thoughts about the notion that they're not off-target effects? This is actually, you are how the body is formed in a dish. And then two, how does that happen? You've probably tried every trick in the book to give yourself pure mesoderm, as the gut people do to give pure endoderm, and these emerge. And they're clearly, the blood vessel, well, those are mesoderm derived, but the CNS, where did that come from?
Yeah, I think that's a very good philosophical question. Fundamentally, when we're looking at these differentiations, it raises questions that we have to resolve with classical embryology. And at some level, we've got to go back to those studies, I think, and just re-examine them and think about, where do these dogmatic principles come from? How do we know there's really ectoderm? How do we know there's mesoderm? Assuming that those principles are rock solid, there must be something different about the pluripotent stem cell differentiation. And maybe these aren't off target. Maybe, in the early days, I thought maybe the brain organoids that were forming next to the kidneys were sending Wnt signals that helped induce them, and that may be the case. So, we know that the kidneys have neurons, for instance. Maybe these are neurons. So we don't know enough about these, quote unquote off target populations. So it's sort of a dark art of human developmental biology that's beginning to emerge out of pluripotent stem cells, and we need to be thinking about this question and be careful with our own natural tendencies to think of a cell as either good or bad, or representing something in the body or not. And I think that's the best course forward, and eventually we'll begin to understand.
Yeah, that's a good answer. I can say, as both a neuroscientist and a major proponent of the school of thought, that the neural crest, which gives rise to the autonomic system, is actually pivotal for not only investing every other organ system, but even directing some of its cytoarchitecture and formation. I actually think it's a fascinating thing that every organ system, not just the kidney. I just came from an intestinal stem cell meeting where they're looking at gut organoids, and the enteric nervous system, again, neural crest derived, seems they also pejoratively use the word contaminate, and one could make the argument actually by necessity in an obligatory way are there in the cultures and they make the cultures work, the organoids work.
And
So could be the same there.
You know, biology has predestined these cell types to arise together for whatever reason, right?
Yeah.
So we have to understand what that is.
Any other questions in the room, philosophical or otherwise? And I see nothing on Zoom. So thank you so much for attending. Thanks, Bino, for educating us. And we'll see you next month for the June installment of the Southern California Stem Cell Consortium.